All right, everybody sees my screen, all right? Yes. Okay. So, hi everyone. As Margaret was saying, I will give you today an introduction to the R bioconductor ecosystem. And the materials for today's session are in this GitHub repository. I think that Margaret and Stephanie share this link with you in the email, but in case you have not downloaded the materials, you can do it by clicking in this link in the presentation, and we'll lead you to the GitHub repository. Or if you want to download the materials directly from your RStudio station, you can run this code and we'll download the full repository with the RStudio project that we will use. Also, the slides are available at this link in case you want to follow these slides. So what is Bioconductor? For those people that have not listened about Bioconductor, Bioconductor is a repository with a huge number of packages. At day, it contains almost 2,000 software packages, and it could be considered as a cousin of CRAN. But the difference versus CRAN is that Bioconductor have packages completely focused on the analysis of genomic data. And what I mean with genomic data, let's say that we are interested in study the cells of the animals or the humans, the plant cells, or even microorganisms such as bacteria or viruses. So the first step for studying these cells is to isolate the genomic material that is contained inside each of the cells of the organisms. And let's think on the genomic material as a giant book with instructions that leads the behavior of each cell in our body. So in the laboratory, we isolate the genomic material. It means this book with instructions, but this book at this point is closed and we need to read it. So we use some specialized machines that are called DNA sequencers. And these machines help us to open this book and read all the information. But the output of these machines are some pages of letters and letters and letters that are not arranged in the properly way. So here is where the power of R and Bioconductor comes into the action. So we use R and Bioconductor and specifically Bioconductor packages to analyze all of these letters and convert this information into a meaningful information and meaningful results. For example, if we are interested in a study two biological conditions, for example, from two patients, one is a healthy patient and one is a patient with certain disease, for example, a tumor cells. So this analysis of genomic data could give us some clues on how the disease is behavior, or how can we treat this patient. And Bioconductor have 4 different types of packages. The first type is the software packages, and these packages contains functions basically. And these functions help us to read the sequences files. I mean, the sequences files are these letters and letters pages. Also have functions to count, normalize, find differences in the genes that are contained in this book, and also plot our results. On the other hand, there are also annotation packages, and these packages are basically databases with sequences of organisms that are well known, for example, the human genome. And also these annotation packages provide the gene identity for all of these letters that are contained in the book. Also, in Bioconductor, there are experiment packages that are a little different from the annotation packages because in the experiment packages, the authors provide the sequences from a biological experiment. For example, let's think that in a hospital, we have a patient we interested in disease, and we want to compare how the cells of this patient are behavioring versus a healthy patient. So these sequences are going to be a little different versus the general sequence of the human genome. So this kind of databases, but with biological meaning, are provided into the experiment packages. Also, this kind of package contains some functions to analyze this specific biological data and sometimes lead to a result with biological relevance. And finally, we have the workflow packages, and these packages are the less abundant in Bioconductor, but are really important because they show the implementation of multiple Bioconductor packages in order to reach some goal. For example, if we are comparing which genes are turned on or turned off in certain biological conditions, we don't only use one package from Bioconductor, we use several packages. So the workflow packages teach us how to implement several packages with a certain purpose and show the steps to perform an analysis. So how do we access to Bioconductor? The first way is entering on the web page that is in this is located on this link. So if we click on this link, we will go to the home page of Bioconductor, and we will see that here are some guidance on how to navigate on Bioconductor, and also here are the place where we can enter to the Bioconductor packages. So for example, let's go to the software packages. And in this page, you will see the list of all the packages available in Bioconductor. And you can search also for a package of interest. Let's look, for example, for the biostrings package that we will use today. So if you go to the biostrings page, you will see all the information about this package. For example, the authors, the maintainer, how to cite this package, and also installation indications that can be run in R to install this package. And more importantly, the documentation. One way to access to the documentation of a package in Bioconductor is using the function browse vignette and the name of the package. And the other way is to click in on the vignettes that are in the web page. For example, here we are going to the PDF, and here is the vignette for this package. And in the vignette, we will see how to use this package and some examples on how the function works. So that's the first way to access to Bioconductor. And the second way is directly from R. So if you run this code in your RStudio session, you will install BioC Manager. And what is BioC Manager? Is a package that helps us to install any package from Bioconductor. In the same way as install.packages install any package from CRAN, BioC Manager and the function install will install any package from Bioconductor. And also BioC Manager helps us to keep all our installed Bioconductor packages up to date. And this is very important because Bioconductor is updated twice a year, around April and October. So I recommend you to keep an eye on the new releases and keep all your packages updated. And this talk is about the bioconductor environment. But why do we need a special environment for working with bioconductor? That's because the data, the biological data, such as the genomic sequences, are treated a little different versus we used to work with lists or data frames. So we need specialized packages to treat with this kind of data. And I will introduce you today to some packages that I found useful to start working in the bioconductor environment. So we will see today these 4 packages and just some introductory functions of all these packages. So the first package is biostrings, and biostrings will help us to manipulate genomic sequences. This package have functions to import and write biological data, such as the FASTA files that contains the sequence of the genes or the genomes, or even the proteins code. So let's open our first script in RStudio. So I will zoom a little. In the repository of the materials, we have different folders, and all the R scripts are in this folder R. And the first script have all the installation indications for the packages that we are using today. So if you want to follow the exercises, I recommend you to install all of these packages. Regu Tools is a package that I will present at the end of the presentation. And this package has several dependencies. So if you have any problem installing this package, don't worry. It's just for an example at the end, but it's not mandatory to have it installed. Oh, sorry. So let's go to the script managing sequences. And let's load the biostring package. And the first thing that we are doing with this package is to read a special kind of object that is defined inside this package biostring, that is a string set object. And we can read a FASTA file that is a very popular kind of file used in the biological research. But you have other options inside this function to use other kind of files if you need to. So the first instruction is to read this file that already is contained in the repository. And if we print this first object, we will see some letters that correspond with some genes from a bacteria that is called E. coli, Escherichia coli. So here are just 5 genes, just for the example. And as we can see, we are just seeing part of the sequence. It means here we see part of the sequence, ... and then other part of the sequence. So it's just giving us an overview of the data in this file, but not all the sequence that will fill all of all our console. And if we ask what what is the class of this object, we will see that it's a DNA string set. It means it's a string set object, but specifically we are saying that these sequences correspond to DNA. And also is a biostrings object because it's declared in the biostrings package. And other things that we can do with this package is to get the length. It means the number of the sequences in this object. For this example, we only have five sequences, so the length of the object will be 5. And also we can see what is the number of characters contained in each of the sequences in this object. So this data were observed also when we print the object here in the width. But in this case, it's easy to see the width of the sequences because they are just a few sequences. They are just 5. But if we are working in the real life, we will have a lot of more sequences. So it's impossible to print all the object and obtain the sequences, the width of the sequences. So that's why we need to use the ensure functions to get a vector that will contain the size or the length of each of the sequences in the object. Also, for biological purposes, it's useful to know the frequency of certain letters in the sequences. For example, the percentage of GC content. So there is a specialized function that is called letters frequency that will allow us to explore how many letters do we have that correspond with specific nucleotides. So if we use letter frequency, we will see the number of nucleotides that correspond with G or with C. Also, we can evaluate just one of these letters, for example, just the G, and we will see the result. Another important thing that we can do with the DNA string set object is to subset sequences. So, for example, if we want to retrieve the first two sequences from the E. coli genome, we can use the square structure, such in the same way as we used to do it with data frames or with vectors. So we will retrieve the first two sequences that we want. But also, in sometimes for biological purposes, we want to extract just a part of the sequence for each of the sequences in the file. So, for example, with subset function, we can retrieve the first 10 nucleotides or the first 10 letters from each sequences. It means this subset will be applied to each of the sequence in the E. coli object. And here is the result. Also, biostrings have some functions to translate the sequences. It means initially we have the DNA code, but we need to translate the DNA, well, we'll transcript, pass it from DNA to RNA, and then the RNA translates into amino acids. So we also have a function to simulate this translation. So we can do it for the full object, but here I am selecting just the first sequence, and I am using the translate function. So we are not seeing anymore the ATCG letters, but we are seeing amino acid scope. Also, there are some times that we need to use the reverse change of the DNA, so we can use the reverse function to get the reverse. Oh, sorry. To get the reverse of the sequence, and also the complement of the sequences. And also you can use reverse complement to get the reverse complement string for each of these sequences too. So there is a lot of functions left in the biostring package, but as I was saying, I'm just giving an introduction. So let's move to the next package. So as you can see in these examples above, we are seeing in the E. coli object just the sequence and the name of the sequence. If you observe here, we are seeing the width of the sequence contained, and the names of the genes in this object. But probably we need more information than just the names, the sequence, and the width. For example, we could need the genomic position and the chromosome where this gene is located. So in the genomes, the genes are usually arranged in this way. We have the full chromosome here in blue. We have two strains, one forward and one reverse. And in the full chromosome, the position of the genes are not all in just one of the strands. So we can have the gene A in the position, it means in the character position, from 10 to 15, for example. And then the gene B in the positions of 20 to 27. And we have another gene that is called the gene C in the reverse chain of the genome. It means the minus strand. And the position of the gene C is in the 32 to 42. So probably we need this information of the gene's position. And if we need that information, we need to create and use a different object that is called the genomic ranges object. So there is a package that is called genomic ranges that includes the definition of a new class of object, the geRanges object. And this kind of object allows to save more information. We still have the gene's name, but also we have the positions, the start and the end position for each of these genes. And we have the strand, plus or minus strand. And if we print this genomic ranges object, we will see that we will get the sec names in the left, then the ranges positions, and the strand of the genes. And if we remember the plot in the last slide, this corresponds with the positions that were shown in the plot. So let's go to the script that is geRanges. And here we can load our genomic ranges package. We will define this object that I was describing, the genomic ranges object, with the set names, the ranges of or the positions of the genes, and the strand. And we will print this object. So here are the information that I was showing in the slides. But note that we are using a special way to define the genomic ranges positions. So we are using this function that is iRanges to define how the positions of the genes are provided. So if we print, just to take a look of how the iRanges object looks, we will see that here is the start that we provided, the end, and the width of the genomic position. Also, we could define this iRanges object by providing just the start and the width for each of the genes. That's another way to define the ranges positions. But also, we can all add more information. For example, I was telling you that the chromosome where the gene is located can be stored also in this genomic ranges object. So let's define another iRanges object, but in this case, the start and the end positions are different. And this object, that is the genomic ranges object, will include a new kind of object that is the RLA object. And this is important because this structure allows us to include some repetitive information. For example, let's say that the genome has 100 of genes. It will be a little difficult to write 100 times the chromosome 2 because all these example genes are located in the same chromosome. So instead of write 100 times the chromosome 2 word, we could use the RLE object, and we are providing the number of repetitions that this character is provided in the object. So the seq names in this case will correspond with chromosomes, and also we are defining the ranges in the same way as we did it before. And also for the strand, we are using, again, this RLA strand. So let's define this new object, the GR object, and let's see how it looks. So if you observe, this GR object looks a little different than the object GR was looking before, the above object here, okay? This is like a basic structure of genomic ranges, but when we add more information, for example, the chromosome and the strands, and also we are adding some metadata columns. It means here we are storing another information that could be important for us, but has nothing to do with the sequence composition. So this object is the result of using the RLE objects in the definition of the genomic ranges object. Also, genomic ranges have some functions to select genomic region. That's because thinking on biological data and biological relevance, we have some composition in the eukaryotic genomes. In a bacterial genome, the genes will be located basically as it's coded in the chromosome. But in the case of eukaryotic genomes, the genes go through a process that is called processing and splicing. And this means that the genes have some regions that are called exons and other regions that are called introns. So when the splicing process goes in the cell, the intron regions are removed from the gene and the exon regions are kept. So at the end, we will get this kind of a structure of our gene. And it's important for biological purposes to know the positions of the introns and how the exons will look like after the processing. So if we if we add the positions of the exons and the introns of some gene in a genomic range subject, it will look like this example. So here we have the seg names that will be the same because we here have the sequence of the first exon. It means this region here. Then we have a gap, basically. Here are from the 15 nucleotide to the 32 nucleotide where there are not sequence, but in the beginning there were a sequence. That was the intron. And then we have the second exon that is here in the right, and it corresponds with the second sequence here in the genomic range. So if we have this kind of a structure, we can see probably we are interested in knowing what is the real range of this gene. I mean, I know that part of the gene goes in this range positions and the other part of the gene is located in this range positions, but I want to know the full range of the gene M. And that's where the genomic ranges package it's important because we can use the range function and let's declare this place. Yes. So here we have the gene M with the two regions, and now we can use the range function, and this will tell us where is the full position of the gene, independently of where the introns and or the exons were located. Also, thinking on biological relevance, there are some genomic regions that act as a regulator on turning on or tuning off the genes depending on different cell processes or cell status. So in biological or in bioinformatics, these regulatory regions are very important and are very studied. So it is important to define where is located this regulatory region. And the flank functions will help us to do that. So let's think that we now have the gene O that has other different positions. In this case, this gene is located in these start positions and these end positions. And the genomic ranges will look like this. So we have the strand, we have the exon IDs, we have the positions, and now we know that the regulatory regions could be in the 100th position upstream of these genes. So the flank function could give us some other positions, giving us the regulatory regions for each of these genes. Look that we have the strand, the start position here, and now is 100 position before. Okay, so that's how the flank function works. In the same way as biostrings, genomic ranges have a lot of more functions, but here I'm just providing an example of some of the introductory functions to see the potential of this package.
Jocelyn, can I ask you a question someone posted in the chat a little while ago? It was actually from the your previous .r file, the managing sequences, about the translate function specifically. And they were wondering about how it handles introns and exons, for example, if you know the answer to that.
Yes, I think you need to provide the processing sequence. I mean, by itself, doesn't distinguish the introns and exons. So you need to use this function after the process of splicing. Yes.
Great, thank you. I think that was the main question that was posted. Are there other, just while we're stopping, do other people have questions at the moment before she goes on to the next section?
Okay.
No, okay. I think we're good to go.
Great. So I was telling you that there are some annotation packages in Bioconductor that contains basically databases. So where can we find genomic data? AnnotationHub is the answer. AnnotationHub is a bioconductor package that helps us to connect with multiple genomic data providers, because we know that there is the UCSC database, there is the Ensemble database, and AnnotationHub helps us to connect with all of them. So first, we need to use the AnnotationHub function to connect with the database. So let's go to the annotation script. And we load the library. It could take a minute. And we will use the annotation hub, and we'll store the result of using this function. It means the result of connecting with the database in an object, just to facilitate the use. So, sorry. This will take a moment too. Okay. So if we print the content of this object, we will see that here shows the snapshot day, that is today. And here is the list of all the data providers that AnnotationHub connects with. Also, here is showing the species available, considering all of these data providers, and the data class of the objects that are contained in these databases. So here we can, once we connected with the database, we can explore these characteristics, for example, the species available. So here we are seeing just the first 10 species available, but if we see the length of all the species available in the databases, we will see that are more than 200 species difference. And for each species, there is more than one data set usually available. For example, for human genomes, there is a lot of information. And also, we can explore which are the providers. So here in genome, oh, sorry. Here is by genome name. So first, I am seeing the genome information. So I am looking for the H gene, the HG pattern, because the human genomes start with these two letters. So here I am seeing all the human genomes versions available in the databases. I think the most recent is the HG38, and one of the most used human genome database is the HG19. And next we can see all the data providers. So as we can see, there is a lot of databases available through the connection with AnnotationHub. And also we can see the class of objects available in these databases. And this is important because it gives us an idea of what kind of objects we will work with. Next, if we want to extract or get one of these genomes available in the database, we need to use the query function. So this function needs the connection with AnnotationHub that is already saved in this object, AH. And also, you need to provide some parameters, for example, where you want to look for the genome, which species, what you are interested in, and which kind of object you expect from the database. So in this case, we are searching on the UCSC database. We are looking for Homo sapiens that is human, and we are looking for genomic ranges objects. And we will store the result in the HG object. Okay, so now we need to use display. So we don't just print the HG object, but because this gives us just some of the information as we were seeing in the AnnotationHub object, but this doesn't give us a lot of information from the real sequences included. So we need to use display. And here, it doesn't seem very well. Okay, I will try to make it a little bigger because we can explore in the console all the parameters included in the query, but also we can search for a certain species or genomes using a Shiny app that is called with the display function. So here we can make some filters in order to get the genome that we are looking for. But if you don't want to display the Shiny app, here you can see some pages. Here is the genome code. Here is the species, the genome name, the provider. So you can explore here in the viewer all the available genomes in the query that you just called above. So once you have the desired genome, it's time to download the data.
Jocelyn, there was a question in the chat.
Yes.
Is there a way to search for species belonging to a specific class or family, like all the mammals or all the primates, for example? Do you know?
No, I don't think so. I don't think so.
Yeah.
Yes.
Thanks for the question.
Yes. What you could do in that case is that separately, you have like a list with all the species names and the property of it is mammal or bacteria or virus. And what you can certainly do is to provide a vector here. Instead of using directly the species name, you could provide a vector with a lot of species names. So if you have already a list into a vector, a list of names, you can retrieve all the species that we want to obtain. That's possible. Yes.
Great. Thanks. Yeah.
So once we have the key of the genome that we want to download, we will use a double square bracket just as the list subsetting, and we will extract from the HG connection this specific genome with this key. So here I will keep. and see that we are now loading the data. So here is where we are accessing already to the genome data. And this could take a moment. Okay. And if we print this object, genes, we will see an overview of the object and also observe that the structure is a genomic ranges object, just like the examples that we were seeing before. So all the functions that are contained in the genomic ranges package can be used in this dataset that we obtained, and this is already the full genome of human. So AnnotationHub, as I was saying, connects with all the data providers for all the species available. But there are also some packages that help us to give a name to each of the genes contained in the genome. So in this point, as you can see, the sequence only contains the chromosome where the gene is located, but doesn't have a recognizable gene name. So we need to use other packages that have this annotation information. It means the gene names. So there is a package that is here, the RHS-AGDB, and this package only have information about human genome. And this package contains all the gene names and makes cross references with the genes keys that are provided in some databases, for example, with the gene bank or other gene identifiers. And how this object looks. This object contains a lot of information, contains the name of the organism. In this case, this corresponds to the human, so it's Homo sapiens. Also gives some information about what kind of keys contains this database. For example, the interest genes, the key genome IDs, the gene ontology assignations. So all of this information is contained in this database, but not the sequences. So the sequences need to be downloaded with AnnotationHub, and then we name the genes with different type of IDs using this specific package. And this also is an annotation package kind. So here we can see all the, Here we can see all the information available for this specific database. For example, the different IDs like the UniProt IDs, the Unigene IDs, the enzymes IDs, ensemble IDs, and this information is available for each of the genes contained in the genome. And now, in order to retrieve some information, we need to know which are the names contained or available for each kind of information. So we use the function keys, and we need to provide the key type that we want to explore. Okay, so here is basically the kind of information that we will find in the column gene name. So it means that for the X object, we have a lot of columns that are these ones. These are the columns, and these are some of the information contained in each of these columns. So the way that we can retrieve some information is by knowing the columns names and some content of these column names. So the columns are the same as the key types. And now, once you have explored a little your column of interest, you can select the gene of interest, and we use the function select to retrieve the information about that specific gene. For this example, I am using the gene name that corresponds with this chromatin remodeler ATRX. So I know that this information of the gene names correspond or is located in the column gene name. And I want to retrieve the interest ID, the gene alias, and the UniProt IDs. So here is the way we can retrieve some information. And the result looks like this. So here is all the result from the select function. I am using the gene name column to make the filter. I am using the gene name, the ATRX chromatin remodeler, and I am retrieving the interest ID, alias, and UniProt IDs corresponding with these genes. Also, you can select multiple keys here just by using concatenate. And as well as with the previous packages, Annotation, DBI, and AnnotationHub have more available functions, but here are just a few of them. And here is just an introduction on how to retrieve some information from the databases. And now another thing that we use a lot is, and the people love it, is to plotting the biological data. So to plotting the biological data, we are using the package Chavez because this package have functions to make visual representations of the genes arrangement in the genomes. And there is other packages that also have functions for this purpose, but the pro of using Chavez is that this package implements a lot of plotting flexibility. So we have a lot of functions to customize, for example, the color of the background, or where is the region that we are interested on, or which are the features, the genomic features that we want to plot. So let's see some examples of how to use this package. And let's go to the number 5, visualization. So first, we will load the package. It's taking a while. Okay, there is. And now we will use some data that is included in the package for examples or exercises. So this data from CTG Islands comes with the Chavez package. So we will load this data, and let's take a look on how this data looks. So as you can see, this object is also a genomic ranges object. And here is the sequence name that corresponds with the chromosome where the genes are located, the genomic position ranges, and the strand. In this case, there is this symbol for those genes that are not known in which strand are located. So in the strand, it could be a plus symbol, a minus symbol, or also this asterisk symbol for the non-known. And we need to use a special object that is called an annotation track object. So we need to convert this genomic ranges object into an annotation track, because this is the kind of object that Chavez is expecting to work with. So here we are just providing the genomic ranges object and one name for the track that we are using for this data. Okay, and as you can see now, only give us some information about the genome, the chromosome, and the number of features that contains this object annotation track, but is not displaying all the information in the same way as the ranges object does. And the plot tracks functions is the main function of the Chavez package. So with plot, we just need to provide this annotation track object, and this will create the plot here in the right. So as you can see here, we are seeing the 10 features contained in the object, but we have no reference about the genomic positions of these genes. So we can add the position reference by calling to genome X extract and store and keep it this genomic X extract in another variable. So now we run again the plot tracks, and note that here we are creating a list listing all the tracks that we want to plot. So here is the genomics X track for reference, and then the annotation track that we defined above. And now we have a position reference here in the top. Okay, so now we can have an idea of where these genes are located in the genome. But it's more helpful to have a visual representation of the full chromosome and see where these genes are located in comparison with the full size of the chromosome. So the chromosome reference is called the chromosome ideogram. And this is a simplified visual representation of the full chromosome. So we need to identify first what is the genome of the data that we are plotting. So we call the genome function to keep this information about the genome. Now we need to keep the seq names of the data that we are plotting here, that from these genes here. And now we need to use the ideogram track function that will annotate all the positions that we are plotting here versus the chromosome size, the full chromosome size. This could take a minute. Right, and we finally plot. Okay, so note that here in the top, now we are seeing the full chromosome, and with the red rectangle, we are seeing where these genes plotted here in the bottom are located. So now we know here this position or these range positions correspond just to this small portion of the chromosome. It's important to keep in mind that the chromosome doesn't have the same scale as the gene's features, because we need to make like a zoom in the interested region where our genes are located. So that just to have this in mind, in consideration. Also, you can add more tracks. The track is this line or this row where is located some of the features, some of the genomic features. So you can add more tracks, but it's important to keep in mind that each track needs to be defined separately. So you need to define one by one the tracks that you want to plot. So here we add a gene model's information. It means that we'll plot all the genes that are available in the chromosome. And also we can plot their gene names. So we need to call the gene models, that is a data set also included in the Chavez package. Here we are calling again a gene region track that will use these gene models data. We will take the genome as the genome corresponding to the genes that we had before, that as we did it here, the genome of the dataset CPG island. And we will use the chromosome names that correspond with this information too. And the name of this track is gene model. This name is the one that is displayed here in the left of the plot. And now you can plot all the tracks. And here they are. So note that we have the information of the genes that we were seeing before, but also we have all the genes structure available in this genomic range that we are observing. And if you need to focus in a specific region of the genome, like making a zoom, you can establish from and to positions to make this plotting specific from a region. So here we are making a zoom and just seeing from this range of genomic positions. So here we only have one of the 10 genes that we were seeing before, and the gene model corresponding to these positions. And as I was saying in the beginning, the pro of using GBs is the plot customization. So we can customize, for example, the colors, because you can see that here the gray panels are probably difficult to see. So probably we want another colors. So we can do that in two ways. You can modify the parameters or the plotting parameters directly when you define the gene region track by adding some available parameters like the background title, and you specify the color in this case that you want to show. So let's see how this looks. And also you can use all the parameters available by using display pairs of the object that is the genomic track. So let's see just a few of them. So for example, you can modify the filling color. You could modify if there is the width that you can be greater or thin, the arrowhead, and there is a lot of parameters to modify for display. And as I was saying, the first way to modify is directly when you define the gene region track And the second way to modify the parameters is by listing the parameters that you want to modify. For example, here we are modifying the color of the background panel that correspond with this white space here in the back of the genes models. So now let's see how the plot results. And here we have the result. We have now a yellow background and some kind of between red or brown background in the track name. That this modification is what we did here. And well, you can explore more about the displaying parameters to modify and customize as much as you want. And you will end with beautiful plots and representations. And also here, note that I included in the parameters modification, I added the transcription annotation, and I established it as showing the symbol of the genes. That's why now we are seeing here the genes symbols or gene keys. That gives us a lot of more information about the context or the genomic context where our genes of interest are located.
I think we're going to pause for just one moment now to see if there are questions. Since we are close to the end of when we normally have the talk part of the session, I just want to give people an opportunity to check in and ask questions if they have them. And otherwise, I'll then let you finish if you would like to. But are there any questions from anybody? People should feel free if you have to go. I understand. We understand that it's like close to the end. Yeah. Someone have a question? All right, I guess not. All right, well then, why don't you go ahead and continue?
Okay, thank you. We are close to the end. Sorry if I am taking more time. So as I was saying, there is a lot of packages available in Bioconductor, but if you still don't find the right package for your data set or for biological purposes, you can also submit your own packages. So I just want to talk you a little about our experience submitting the RecuTools package. This package borns with the enthusiasm of the community of software developers for bioinformatics. That is the CDSB for the letters in Spanish. And probably Leonardo have told you about this community before. This community is focused on increase the number of software developers, especially focused on the R language in Mexico and Latin America. So here is the RecuTools is the first package that we have submitted to Bioconductor, but we hope that there are many more. And RecuTools borns because there exists a database that is called the RegulonDB database. And this database contains information about a bacteria that is called the Escherichia coli. And this database is automatic and manually curated. And there has been a lot of researchers working in this database for almost 30 years. So it has a lot of information about the genomic regulation of this bacteria, but this database is provided in a SQL format. So we wanted to provide a tool that could facilitate the access to this database and do it by the R and Bioconductor ecosystem. So in this package, we included some functions to connect with the database in the similar way as we see the connection with AnnotationHub. So we have functions to connect with the database RegulonDB. Also explore the database, it means explore the datasets contained, and also subset or subset some database of interest or filtering, and also to plot some of these data, all of these data sets. So how Regu tools work? As I was saying, first we need to connect with the database. Then we create a new defined object that is called a RegulonDB object. And we defined this special object because we wanted to keep the organism name, the data version number, and the genome version number, because in this moment, the RegulonDB only have information about this bacteria, E. coli, but the scientists are working on creating multi-organisms versions of this database. So that's the importance of keeping all of this metadata in the object. And then we can use some functions created in this package to filter the information from the database. So in the same way as we were seeing before, we need to provide the data set that we are interested. In this case, we are interested in the dataset gene that contains all the genes information, such as the gene names, genes positions, some bibliographic references, etc. We want to retrieve some attributes, for example, the positions in the left and the positions in the right, the strand and the gene names. And we can use some filters, for example, only to select some genes that we are interested on. And also, we integrated this RegulonDB object with the Bioconductor ecosystem. So we integrated one function that allows to convert the RegulonDB result into a GRanges result. So you now can see the GRanges object that we are now familiar with. Also, we integrated some functions from the GBIS package to visualize the positions of these genes of interest. By now, we didn't include the chromosome representation, but we are working on do that. That's because we have a little different definitions on the genes and the references of the genes provided in the RegulonDB database. So we need to make it match with the UCSC genomes references genes to include the chromosome visualization in the top. So that was a beautiful experience to create it, writing, and submitting our own package in Bioconductor. And there was a lot of feedback from the main maintainers from Bioconductor. So if you want to submit your own package, I encourage you to do it because it's an excellent and exciting experience. You can download this package from Bioconductor too by using BioC Manager or install Regu tools. as well as we did it with biostrings or genomic ranges packages. And where can we find some help when we are using bioconductor packages? As I was saying in the beginning, exists the browse vignettes functions, and also in the web page of Bioconductor is located all the manuals for each package. So that is a good start point, but it's good to know that the bioconductor community is an infinite source of knowledge, and it's a community very kind and warming. So don't be afraid to ask for some help. And Bioconductor have a support page that is located in this link. So if you go to this link, you can see all the questions that the community is asking. And here you can ask directly to the authors or the maintainers of the packages for a problem. For example, you are experiencing through some issue with your data set and you have no clue of why it's failing, you can contact directly to the maintainer and ask for some help to resolve your problem. Also, there is a Slack channel from the Bioconductor community. You can join to this Slack channel by clicking this link. And there are a lot of spaces and different subjects. And there is also a developers forum. So if you go beyond and start creating your own packages, probably you are interested in following the developers forum. And also the developers forum have monthly sessions, meetings, and where you can present, you can show your contributions and hear from other developers and learn a lot from them. And also share your contributions with the community. It's very important to learn what the other people are doing and what are the new packages that are developing. So thanks to a developer scholarship, I had the opportunity to go to the Bioconductor conference in 2019 that was held in New York and the Rockefeller University. And I presented the advance of the RecuTools package. At this point, the RecuTools package was not finished, but presenting my work in the conference was very helpful because I had some feedback that will lead us to improve our functions and add some additional functions too. And also knowing the community is very excited, and I was very happy to meet a lot of incredible people that I was just knowing by their names in the Bioconductors authors' names, but meeting them in person was a beautiful experience. And due to the pandemic, this year, the Bioconductor conference, as well as in 2020, will be held online in a virtual mode. So all the registration for the conference is already open, and you can register for the conference in this link. And that's all. Thank you for your attention, and thank you for the invitation. If you have more questions, you can contact me by Twitter or by my GitHub or by email that is located in my webpage too.
Wonderful. Thank you so much for your presentation. It was really a nice overview of a lot of the things that Bioconductor has to offer. It's a huge amount of packages and materials, and I think you hit on some of the key highlights for sure. I'm a huge user of Bioconductor myself, so I know I can appreciate what a monumental task it is to summarize it. So thank you for that. I think it was really nicely done.
It was my pleasure.
Yes, I'm so glad that you could join us. Are there other questions that people have? So there was one question about one of your data visualizations, where it was a little unclear if you were looking, there was a CPGs object or a data set, and then there's also a genes data set. And so that final plot that you showed for that one, was it multiple tracks that showed the CPG islands and then also the gene annotation in the same region that you were looking at? Is that what it was? I can't remember.
Yes.
Yeah.
Yes,
okay.
it was the location of the CPG islands in the first track in the top. And then it was the full genes locations in the second track. Yes.
Okay, great. Thank you for clarifying that. Are there other questions before we wrap up this part? I especially did really like your shout out to the process of submitting a package to Bioconductor. I know people who review packages for Bioconductor, and they do take it very seriously, and they try to make sure that the packages that are on Bioconductor, so it's a whole nother level of quality that you get compared to even CRAN or something like that, right? So the packages get really thoroughly reviewed for having good documentation and meeting sort of a specific set of standards in terms of how they're coded and that kind of thing. So you know that when you use a package from Bioconductor, that it's gone through a series of quality checks that will make it so that you have more confidence in the package and in its ability to do what you want it to do. So yeah, so that was definitely, I'm glad that you've had a good experience going through that process as well with your own package.
Yes, definitely was a very good experience, and I learned a lot from that.
All right, well, I'll stop the recording, and then if people want to stick around, if they have other